everyone, and welcome to today's class on hemodynamics. We're going to be going through the different technology comparisons of available monitoring techniques within the market. The first central question is, why do we monitor? So we rely on the clinical status of a patient to understand how they're doing hemodynamically. However, there are monitoring techniques that we can rely on to gain specific insight into parameters that we may otherwise not visibly see. When we seek to find the pressure and oxygenation, we may rely on, specifically in the context of an ICU or an OR, on pulmonary artery catheters. They are the gold standard. They are extremely invasive, and this is why we reserve this to very severe cases. We basically need a central access, so either a jugular vein or a subclavian vein that will migrate to the right cardiac chambers and allow us to understand how a patient's saturation is doing intracardiacally. In terms of the volume and lung water, this is going to be one of the parameters that we can gain insight into a patient's fluid status or preload, and we're going to rely on transpulmonary thermodilution. Some of these, the way in which it works is we inject a solute that has a specific temperature, and through the measurement of the gradient of change in the temperature, we can estimate the volume that the patient has. This is also done not only through temperature changes, but through the rate of dilution of, for example, lithium in specifically transpulmonary thermodilution. However, this is limited to an extent, as we cannot indefinitely inject a patient with lithium to understand and see the degree of change that the concentration of lithium has. And it's also obviously contraindicated in several patients. To understand a patient's structure and function, we can use point-of-care ultrasound, echocardiography. Obviously, these are going to be operator dependent. We could also do more expensive techniques such as a functional MRI or MRIs in general in order to see a patient's ventricular function, valves, and if a patient is suffering for cardiac tamponade, echocardiography would be very helpful. This is going to be a diagnostic supplement, but we will require a licensed physician and a technical expert in ultrasounds to be present to conduct this, and obviously it cannot be done in a continuous manner. In terms of the flow and dynamics, this is a more up-and-coming field, which is composed of pulse contour, which is minimally invasive as it requires only an arterial line, and the completely non-invasive electrocardiometry and bioreactance. I should also mention that obviously echocardiography and point-of-care ultrasound are completely non-invasive options and are very valuable in a clinical context. Over here we see a little quick reminder from past classes in which the different variables that we can gain insight into in a patient's hemodynamic status will directly reflect what the treatment that they need is, in a way that is a lot more tailored, and that doesn't imply using, for example, inotropes if a patient is only suffering from a volume issue, for example, which sometimes the treatment algorithm that we follow is very broad in the pharmaceuticals that we prescribe, and this can allow us to be a little bit more tailored and see the response that a patient has to specific treatments over time. So some of the key points that we're going to go through today are some of the advantages and disadvantages of the available technology. The invasive methods such as pulmonary artery catheters and transpulmonary thermodilution are irreplaceable. They are gold standards. They're going to provide us a right heart assessment and a very reliable lung water parameters. Of course, in an invasive context, this is why we're looking for more options to evaluate patients in non-invasive ways. In the case of pulse contour analysis, they are going to require peripheral arterial line, so they are significantly less invasive than a central line, for example. However, the devices do need to be calibrated, and uncalibrated devices have a very high margin of error, and therefore it's important to remember that it's unacceptable for a device that is measuring a patient's hemodynamic status to have an error exceeding 30%, which some of the uncalibrated devices do. In terms of conventional non-invasive options, we can rely on electrocardiometry, bioreactance. However, the ones that rely on pulse contour analysis, for example, they may be a little bit more unreliable when there's vasoconstriction, in the case of bioreactance and bioimpedance. When there's motion, they may give intermittent answers, and if anything is disrupted within the signal capturing, we're going to not be able to gain insight into a patient's hemodynamic status in that moment. This is why invasive and non-invasive techniques, the minimally invasive ones that need to be calibrated, uncalibrated, we really have to understand what the advantages and limitations are of each one of these so that we can integrate them and make sure that we provide the best patient care possible. Therefore, as we can see in bold, the optimal strategy is to approach this through a multimodal approach and also a phase-dependent approach. It's also going to depend the hemodynamic status of a patient in that moment, the type of monitoring that we are going to recur to. And we should combine different pressure targets, perfusion markers, as well as dynamic measures when possible to see how a patient is doing over time, how they're responding to treatment, and not fixate on one method of hemodynamic monitoring, for example. Over here we can see the invasive reference standards, which we've discussed before, such as pulmonary artery catheter and transpulmonary thermodilution. Obviously, these both have strengths. The pulmonary artery catheter is the gold standard, and it's usually used in complex shocks, in any right heart pathology, where we can really understand the physiology of the right heart by placing the catheter within the cardiac chamber. And it's important to remember that the only technology that directly measures mixed venous oxygen saturation is going to be the pulmonary artery catheter. So in the scenarios in which a patient's in which this parameter is very important to evaluate in a patient, we cannot attempt to apply a different technique. In terms of transpulmonary thermodilution, this also is a strong reference standard, and it's the only bedside technology that reliably estimates extravascular lung water. So again, a little bit of a note over here because the icon does have the parameter of thoracic fluid content, which has been validated. However, we cannot completely migrate into that, and obviously the answer that we gauge from transpulmonary thermodilution will be a lot more reliable. Obviously, these both are very invasive. They require central access, and transpulmonary thermodilution requires both central and arterial access. It's also going to require recalibration, and obviously it's going to limit patient mobility, and it's going to have different impacts regarding the invasiveness of the procedure. In terms of the pulmonary artery catheter, how we have discussed this in classes before, where the pressure in the right heart is usually extrapolated to the pressure in the left, and that is not necessarily true in patients, for example, with left heart pathology. And it can be dangerous to rely on these parameters as fact when we need to understand the physiology behind what we're measuring in order to reliably understand the patient's status. In terms of pulmonary artery catheters, the limitations, as I've mentioned, it's that it requires a highly specialized care and training. It's very invasive, and it has been associated to very severe complications such as dysrhythmias, pulmonary artery rupture, thrombosis, as well as pneumothorax. And in terms of the reliability of the preload estimation, it assumes that the pulmonary wedge pressure is going to be equal to the right heart pressure, and of course, this may not be the case in mitral disease or different conditions. In terms of the calibrated pulse contour systems, this is going to be PICO and LITCO. These are still very invasive. They require central lines and also an arterial line. And they are going to be beneficial because we can gain a continuous and calibrated cardiac output, as well as preload measurements and lung water measurements in the case of PICO. And in the case of LITCO, which is what I was referring to earlier with the lithium diffusion, the benefit to LITCO, for example, is that you avoid thermodilution, and therefore you don't always require a central line. However, it is still invasive as it requires an arterial access, and you also have the added risk of injecting lithium into a patient. Obviously, repeated lithium dosage is not going to be desirable in some patients, and it's going to require recalibration when there are changes in systemic vascular resistance. In the case of the limitations for PICO, it is going to require central venous and arterial line, and it will also require periodic recalibrations. Therefore, it's important to understand that even though these are going to provide us a continuous cardiac output, we may not want to have such an invasive form of monitoring on a patient right off the bat. In terms of the transpulmonary thermodilution, the limitations of calibrated pulse contours, which we've already mentioned, of course, are that they require calibration, they require central and peripheral access, and it's interesting because within these methods, the reading accuracy is also going to be severely decreased. The reliability is going to be severely decreased in the case of hemodynamic instability, and therefore this is a major red flag if we're using these forms of monitoring within patients that are hemodynamically unstable. In terms of the uncalibrated pulse contour systems available in the market, we have the FlowTrack by Edwards, the MostCare by Pram, and the ProIQT by Getinge. And of course, the benefits to these are that they don't require calibration, and therefore they're more reliable when used by different types of personnel that may not necessarily be licensed or particularly well versed in the use of these methods. And even though the benefit is that they are less invasive, what I was mentioning earlier is that the accuracy falls when the patients are most hemodynamically unstable. The errors in unstable patients exceed around 30%, and that is not acceptable within a clinical context. As I've mentioned, the accuracy degrades when the patients are sickest, and they are going to require an arterial line with good signal quality. Another thing to take into account is that usually in a patient that is suffering from shock, for example, you're not going to have good peripheral perfusion, and this is going to affect how readable the hemodynamic parameters you're extrapolating from this arterial access are. This obviously just adds into the high degree of error when patients are hemodynamically unstable, and the fact that it's uncalibrated also makes it unreliable to use as a standalone monitoring technique. However, obviously, as I've mentioned, all of these have benefits. They are responsive, they're dynamic, and they can be beneficial. Furthermore, in the completely non-invasive options, we can rely on point-of-care ultrasound or esophageal Doppler signals, which are going to give us an image of how a patient is doing hemodynamically. Of course, the limitations that we've always discussed still stand. They are extremely highly user-dependent, and in the case of the pressure signals, such as a volume clamp finger, even though it's fully non-invasive, and it's going to provide us with a continuous stroke volume and cardiac output, it's going to be very unreliable in patients suffering from shock or severe hypoconstriction or hypothermia, because of the same reason that we've discussed prior, where if you're not getting a good signal peripherally because a patient's hemodynamic status has declined, whatever parameters that you're gauging from that point are not going to be reliable. It also, the applicability or the reliability degrades when there's a use of high-dose vasopressors, because peripherally the patients are going to be highly constricted. And this form of the volume clamp finger cuff, it's not validated in pediatric patients, as well as the fact that it's very expensive. In this slide, we're going to go through the different limitations of the non-invasive alternatives. In summary, these are the pulse oximetry-derived indices, what we've already discussed, point-of-care ultrasound and echocardiography, the pulse wave transit time devices, which have a decreased absolute accuracy, and their performance does degrade with the presence of arrhythmias as well as rapid changes in vascular tone. Therefore, patients, for example, with shock are not going to be able to benefit from this sort of monitoring. In terms of bioimpedance and bioreactance has been left to the side with the growth of bioimpedance because the technology based on bioreactance doesn't accurately disclose the physical principles it relies on. And therefore, bioimpedance has been growing a lot more. Some of the limitations are obviously that it's going to be a little less reliable. Well, it's going to be less reliable than thermodilution. The signal quality will be degraded by motion, arrhythmias, electrical interference, obesity, and even though in the limitations it also includes the presence of pleural effusion and pulmonary edema, the icon does have the thoracic fluid content parameter, which is going to account for this increase in volume in the thoracic chambers, and it's not necessarily going to be an obstacle for interpreting a patient's hemodynamic status. In the case of bioreactance, the error margin exceeded 30%, and therefore it's, as we've mentioned, which compares to, for example, uncalibrated technology of pulse wave analysis, unacceptable for clinical use. It's too high for it to be reliably used. Now we're going to focus on electrocardiometry, some of the fundamentals. We discussed in the first class the physical principles of bioimpedance, for example, such as the alienation of red blood cells, how during systole, when they all become they all are parallel and become alienated, this is going to decrease the impedance, and therefore we can interpret those signals mathematically and understand when a patient is doing in systole versus in diastole. We won't go through that again. I can refer you to the first class where that was discussed, just for the sake of time. However, I did want to mention that the sensor placement is highly important. It's very important to adequately place the sensors because incorrect placement is going to lead to incorrect signals. As you can see over here, there's 4 sensors from A to D. A is the black sensor, B is the white sensor, C is the red sensor, and D is the green sensor, and they are placed alongside the body. The parameters that we're going to measure through electrocardiometry are going to be cardiac output, stroke volume. We know this very well, we've been through it 1,000 times, and how the different changes we can interpret to understand if a patient needs help in contractility, how their fluid tolerance is. Over here, I also placed a picture on how the electrodes should be placed in a neonate, because one of the advantages of electrocardiometry is the fact that it can be used in pediatric patients as well as infants or newborn babies. Over here, we can see how the different technologies compare to electrocardiometry. As you can see, the measurements where we can obtain a continuous cardiac output, we're going to find this in all of the technologies, while the benefit of electrocardiometry is that it's non-invasive. It can be used in pediatric patients. It does not require calibration, such as compared to the invasive mechanisms, for example, of pulmonary artery catheter and transpulmonary thermodilution. It has very low operator dependence, and it has a very low setup time as well. Therefore, of course, these are some of the advantages as compared to different technologies. In terms of the limitations of electrocardiometry, as we were previously discussing, the signal quality can be severely affected by motion or any electrical interference, as well as certain pathologies, specifically those that are associated with arrhythmias or any sort of electrical change within the heart. It's important to take into account that electrocardiometry has been validated in pediatric patients as well as patient transport. And in the case of shock, it's suggested that it's very helpful by certain physicians. However, it needs to be validated by phenotype. Not every single phenotype is going to be adequately measured with electrocardiometry. Therefore, you have to make sure that it's tailored to specific patients. It's important to know that electrocardiometry under no circumstance is going to provide a structural assessment of the patient, and echocardiography remains required to evaluate a patient's valve status, ventricular function, as well as, for example, cardiac tamponade. Electrocardiometry is also not going to provide us a mixed venous oxygen saturation signal, and for that specific parameter, we're still going to require the use of pulmonary artery catheters. And finally, it's not going to give us, even though, as I previously mentioned, TFC is a value that is going to provide us a rough estimate of the intrathoracic volume content, it's not the same as what we gain through transpulmonary thermodilution. Essentially, TFC is a global parameter and it's a non-specific index for thoracic fluid. It's going to go up with pulmonary edema, however, it's not going to be specific, as it's also going to go up with pleural effusions and hypervolemia, and also patients that have obesity, for example. And the special thing about the extravascular lung water is that it separates obviously between intravascular and extravascular compartments, and it's going to be very helpful in patients with acute respiratory distress syndrome, where the central question is how much extravascular lung water there is. So it's not going to be a substitute, however, it's going to be helpful for evaluating trends in intrathoracic fluid content, seeing how a patient evolves over time, which leads me to one of the strongest features of electrocardiometry, which is the ability to generate a hemodynamic status report that we can divide and see pre-treatment and post-treatment, while also being able to follow a patient throughout the treatment course. And yeah, those are some of the advantages and disadvantages between the different technological comparisons. Thank you so much for your time. I hope this class was helpful. If you have any questions at all, don't hesitate to reach out. And thank you so much.